Welcome to AP Bio Review. We are going to be doing molecular genetics. My name is Aeri. Off camera will be the ever-present Nick, and we are excited that you are joining us, and we're going to do some exciting free response questions together on this particular topic. So we are doing some transcription, translation. I'm going to get you to do some drawings. So lots of questions here about what is covered. The AP, the College Board has said it's the first 6 units for AP Bio. So we're not doing, you're not going to be tested on behavior, animal behavior, ecology, things like that. And I believe, oh, and somebody asked about photosynthesis, cellular. We did some questions on that yesterday, actually. So check out the video recordings if you on the YouTube page, and then you can see what questions that we did cover yesterday. Yeah, Manel, thank you. Yeah, we already did it, but check out those videos. Hey, and if you are new and this is the first time you've watched the stream live, welcome. I'm always excited to see new people. And so I hope you're ready to put on your bio hats and bio glasses, and you're ready to tackle some free response questions. The way we're going to do these is to give you a stimulus and give you some time to kind of work through it on your own. I do recommend you grab some paper and something handy for you to draw and write down your responses. And then once the time is up, I always encourage students to kind of like type in their answers into the chat, but don't hit enter. I know sometimes it's kind of easy to automatically hit enter, so it's totally okay if you do by mistake. But what we'd like to do is like to see everyone's response at the same time, so we don't have any spoilers. But yeah, hi, I'm so glad you guys like these. I'm gonna admit that people are noticing, and we might just get enough incentive to like do more of these. So if you do want to see more, please do let us know that you want to see more by telling your friends to come and watch and contribute and pay attention. Like, I mean, video recordings are always great, but live streams are a way for you to get like your questions answered, to have a response, for me to like, follow along with what you guys have questions about, so that I can modify them for future videos. Because today's lesson was pretty much entirely guided by the question I asked at the end of Tuesday. On Tuesday, I was like, what do you guys want to cover? And so many of you said translation, transcription, those questions about Operons, biotech. And so I try to make sure that the questions we're doing today are geared in that direction. So if you want more, let us know by commenting. And that way I can figure out exactly what you guys need for your AP Bio exams. I'm so glad. I am glad, they're so fun. Okay, bio hat on. I'm gonna start my screen share. Let me know when you see that big old purple. Okay, so molecular genetics, transcription, translation, DNA replication, many different components. The AP Bio, the College Board has said that you're not going to have to know a lot of the intricate details, but really, especially in the consideration that this is going to be a 45-minute free response exam, it's going to be mostly about the application of this material in new scenarios, new living systems, anything like that. So I really don't want you to focus so much on just like vast amounts of memorization, but more about just getting your hand in as much practice as you can. So thank you for the comments. And I see you guys, it is definitely very purple, isn't it? Okay, so let's go real quick. If you've never been to one of these are some of the notes that we have for the new test. It will be at home, 45 minutes, free response only. And there'll be a couple of different dates, but all of the details beyond what is on this page is going to be released April 3rd. So next Friday, you got to make sure that you're checking the College Board site, and they're going to release a lot more details about what is specifically going to be on this test, so that you have a proper amount of time to prep for this. Okay, so let's get going. So just like I always do, I will show you a stimulus. This one's going to be a drawing one, so I'm kind of interested to see how we're going to decide on responses. It might be mostly just like I'm gonna draw it out and you're gonna compare your notes. But I think what I would like for you to do is I'm gonna give you 3 minutes to read the stimulus, take a look at the question being asked, and then write down. And then I will throw in a question into the chat, and then I want you to type in your response. But again, don't hit enter, and then we'll see how everyone does. So 3 minutes for this very first question. Okay, so go ahead and type in your responses for how many double-stranded DNA molecules that you end up with in generation 3 according to your drawing. If you did this properly, you will see, and you can just count the number. And so go ahead and throw that into the chat. So this is a Meselson stall experiment that essentially proved that DNA is replicated in a semi-conservative model. What that means is that every strand of double-stranded DNA is going to be one strand of the previous parent and one brand new strand. And nice, I see the responses coming in. Ananya, Jason, Ariba, yeah, Ben, and oh, nice, well done. And Flora, exactly, that's a great way to specifically show exactly what's going on. 16 strands total, 8 double strands. You guys are all on this. I'm impressed, well done. Okay, so, I mean, essentially this is just replicating that very first experiment. They didn't have a way to visualize it. What they did is essentially, they use centrifugation and they spun it down because, you know, heavy nitrogen is heavier, it'll sink to the bottom. So the ones that had heavier nitrogen would be like a band on the bottom, and then the ones that were grown in nitrogen 14 were kind of a band on the top, and they measure the relative ratio of them all to essentially prove this is what was going on. Nice. Okay, so this is what I'm going to do. I'm going to draw out what your drawing should roughly look like. So real quick, right? So I get 2 strands like this, and then this guy and this guy should be from the parent strand. And then I get 2 more double strands in there, and then this strand, and then 1, 2, 3, 4, 5, 6, 7, 8, and then this guy and this guy. So another variation of a question like this is, instead of asking you to draw it, would be to ask you about like how many of them. So it'd be like 2 out of 8, so a quarter of the double-stranded DNA will be labeled with N15. So that's another variation of the same kind of question. I would encourage you to just do diagrams to show exactly what it is, then you're not going to make any mistakes in the math. Yeah, so the question does specifically ask about double strands. So you want to make sure that, you know, you kind of have them paired. I feel like, you know, for a question like this, they're going to be a little bit nitpicky. Make sure that you're not just drawing 16 strands like straight and evenly spaced across, that you do have to show that they are somehow paired somehow. So like, you know, cluster them so that they're closer together. Yeah, well done. Excellent. Well done. Yeah, I'm impressed. So, you know, the reason why I thought that a question like this would be useful, maybe not exactly the same as the original Meselson stall experiment, would be because of the nature of the free response questions, and honestly, to avoid some of the cheating that can happen. It's not something that's easily search upable, right? So, you know, having to physically draw it, having to like have conditions that are specific to the questions will make it a lot more specific to the situation and a lot harder to cheat on, right? No, it does not, Allison. It totally does not matter as long as 2 out of the 8 double strands have some label on it, yeah. So E. coli is the bacteria that we're using, the student is using to essentially mimic the Meselson stall experiment. It's just a bacteria that they're using as a model system. Yep. Okay, that's it. I mean, that's the entire crux of this question. Awesome job, you guys rocked it. Okay, so let's move on and take a look at our next one. Okay, so just like before, I will give you 3 minutes to work through this one on your own. The question at the bottom is a fair bit longer. There is a little bit of data to analyze. So we'll give you, again, another 3 minutes to do this. Go ahead and type in your responses into the chat. Don't hit enter. And when we come back, we'll discuss this together. So I'll see you back in 3 minutes. Okay, welcome back. Go ahead and throw in your answers to what you think is going on with this gene that is targeted by the microRNA. Hey, perplex. So UCP1 is a really cool protein. It actually stands for uncoupled protein 1 because that's exactly what it does. It uncouples the ETC with oxidative phosphorylation. It is something that is actually endogenous to us. If you were watching in a previous session, we talked about how we, people used to use DNP, dinitriphenol, as an uncoupler, and it did bad things, right? Other than help you lose weight, it'll also kill you if you're not careful. Did you know that we actually have an uncoupler endogenous to us? We actually code for this. And there's a lot of like future research interest into like maybe possibly exploring the usage of UCP1 to help, people lose weight. But in any case, it's not the focus of this one. It's just a cool type protein. But yeah, let's see what you guys are all doing. So Ben says a possible role of microRNA1 is that it targets for a co-repressor. It would allow UCP1 to be transcribed, translated without inhibition. Hey, I like it. I like the word repressor. I think that's an excellent usage of the data that's showing up in that table. So Ephraim has gone in a little bit more directly and says the microRNA itself might be acting as an inhibitor of this operon. So you got to be careful here. We are talking about a eukaryotic system. So, humans, eukaryotes don't use operons. Operons are bacterial specific, but I like the intent of the inhibition that's kind of happening here. Yeah, some kind of inhibitor attacks the gene that it does not attack UCP1. I like again that relationship between the gene targeted by the microRNA1 that is essentially just somehow turning off the UCP1 either by directly binding to the protein or maybe some kind of transcription factor like a repressor of some sort. So yeah. MicroRNAs, what are they? Well, I mean, it doesn't say anything more than it is complementary. So let's think about this. Let's think about this. If we're told that it stops the production of specific proteins by blocking translation. Micro just means mini, like a little piece of RNA that is then binding complementary to other RNA. So what do you think? What is necessary about RNA in order for translation to occur? If you know the mechanism by which translation occurs, you got your big old like ribosomes kind of sandwiching your RNA. But yeah, RNA is actually needs to be single-stranded. If you have double-stranded RNA, the nucleotides are not accessible by the tRNAs to come in and then complement to it and then have the little amino acid there to be able to form the protein. So by microRNAs being complementary and thus becoming double-stranded RNA, it blocks the translation. That's exactly what's going on. Now, you don't really need it. All you need to know is understand that it blocks the translation of protein, okay? So brown one, let's talk about this. Let's talk about this. And it's not that it's stopping the infants. All they're doing is they just want to know more about UCP1. They're not trying to block this in infants. That's just like kind of the context, the background behind what UCP1 is in your body. Yeah, it's okay. So microRNAs is repressing the expression of other genes to allow for more UCP1. That's exactly a great translation of the information given in that table brown one. So yeah. Isabel, if you know siRNAs, it's essentially the same sort of mechanism. Yeah, siRNAs, microRNAs, same thing. Yeah, so usually microRNAs are exogenously done. So it's a way of controlling for gene expression that scientists use. They're like, okay, we've got this gene that's complementary. I'm going to throw it in and see what happens, essentially. It is a hard question, Matthew. You could have Nick to thank for this one. But yeah, it's a difficult one. But what I want you to do is don't get lost. Don't get stuck. You look at the data and you essentially put it in word form. When I block the translation of the gene that is binding to microRNA1, I see lots of UCP1. So maybe what I'll do is I'll draw two genes, okay? So I've got, this is, we'll call this gene 1, right? And that binds to, okay, this is microRNA1, okay? And then I've got gene 2. And that binds to another different microRNA. This is microRNA2, two separate genes. So if you think about this, well, normally this gene, normally in the absence of a microRNA, will get translated into protein 1. Okay, and then when we see that translation of the protein 1 happening, when we have that protein 1 being translated, this leads to a decrease in UCP1, CP1 protein, okay? Somehow. We don't quite know the mechanism by which, but this guy, when we translate this into protein product 2, this doesn't have that decrease. It doesn't really do anything, okay? Protein 2 doesn't really do anything. Yeah, so we don't really understand exactly how that's occurring. So what the question is asking you to do is propose a role. So we had some really good responses a little bit earlier about possibly something that the protein 1 is doing is acting as a repressor. So maybe it's a transcription factor that binds so that you're blocking the promotion of like the promoter site, and then it's not allowing for the transcription and translation of UCP1. That's one method. Secondly, the other way you can do is maybe it codes for a protein, and the protein itself, protein 1, binds to UCP1, and then something else occurs so that you're not getting the translation of that UCP1. Okay. Okay, so that's what I said as my possible answer for this question. So I'm going to go ahead and throw that in here so that you can take a look. And so these are some proposed ways. This is not necessarily the only one and only way to read about this, but this would be a logical conclusion that would support the idea that protein 1 is involved in in repressing the translation of UCP1. Exactly, Sadie, exactly. Well done, okay? So if you take a look, I think, okay, so maybe we'll back up and take a look at the data, okay? We take a look at the data. It says, when I have microRNA 1 there, it blocks gene 1, whatever, I'm going to just call it gene 1. And then all of a sudden, I see a huge increase in UCP1 expression. Okay. And then for microRNA 2, if I block gene 2, I have low levels of UCP1 expression. And how do I know microRNA 2 does not do anything? How do I know that? Like, what is the data telling us? How do I know? What do you think? So I'm thinking that since there's no microRNA here, then the numbers look just the same as microRNA 2. Okay, yeah, so since that number is the same as this and as this, then we can say that microRNA 2 doesn't really do anything, okay? Our microRNA 2 is targeting a gene that doesn't really do anything. Yeah, so that's what you want to do, is focus on the data, focus on the way that these numbers are arranged, and hone in on exactly the changes that are being made here. So in terms of somebody who's asking me about some of the terms, then I would say something like repressor or inhibitor will be something that's necessary so that you can see the differences in this. Okay, well done, team, well done. Okay, we got another question based on what we had seen with this prompt. So I'm going to go ahead and give you a minute and a half for this next question, and I'll see you back in a little bit. Okay, I kind of gave away the answer for B, but go ahead and tell me, what do you think about the answer for our second prompt? So it's asking about the role of running the experiment in the absence of microRNA and explain how it connects to the idea with microRNA 2 and no microRNA. So hopefully the early description kind of gave it away, but you know, I think it's an important understanding behind like things like the experimental design, which tends to be a pretty common question. Awesome, no microRNA, there's no repressor, no inhibiting, means the level, let's be careful here, let's be careful. So notice that the data tells us in the absence of microRNA, we have pretty low levels of UCP1. So that's kind of like the natural state. The natural state of this is that you don't really have any microRNA, because no microRNA means it's like the control experiment. I think that's what I would look for, right? I would look for the fact that no microRNA is a control. So it establishes like a baseline. And the connection that you're looking for has to do with the fact that the number for microRNA 2 is very similar. So really what this is saying is that you should understand that microRNA 2 is, it's binding to a gene that doesn't really affect the levels of translation for UCP1. Yeah, I hear you, I hear you. So maybe what I'm gonna do is give you a chance to kind of read through my written explanation for this. And then I'll try to describe it again one more time. Here we go. Okay. So running this experiment in the absence of microRNA establishes a baseline for how UCP1 is expressed. So it's acting as a negative control. Since the levels of it between microRNA 2 and no microRNA are the same, we can conclude that the gene that binds to microRNA 2 does not affect the UCP1 translation. For sure. And I can see that some of you got it. Some of you guys found this kind of challenging. And it is a very challenging question. I'm not going to lie. This is a very challenging question. But I want you to think about the pathway. What we're doing is trying to figure out what is controlling for UCP1 translation. We're interested in trying to turn it on. The natural state of this protein is off. Like not all genes that are coded for in our genome is always on. A lot of times they were off, right? That's why your skin cell is not the same as your hair cell. It's not the same as your eye cell. There's different levels of translation and regulation in that. What we're seeing here is that by blocking the translation of another gene and looking for its effect on UCP1 levels, then we can see like, how is this gene turned on and off? How is UCP1 turned on and off? Oh, great question, Anika. Does the 0.5% lower expression of microRNA 2 have any significance? Now, we're not given any measures of significance in here. We don't know that. But you know, you have to kind of be able to eyeball it. Notice how huge the microRNA 1 levels are compared to the other two values. And then you can roughly conclude that as a trend, those two levels are around the same. Yeah, and I would mention that it is a negative control. I think that's an important component of identifying the parts of an experiment. So saying for sure that's a negative control. Good question. What is the difference? And Ellie asked about positive and negative. So positive control will be like a protein you know would be a repressor. And so if you got rid of it and you saw the levels, then that would be a positive control. Like you know the outcome of it. So if I had another gene, for sure that would act as a repressor, and then I also had that in this experiment, I can also compare the values of what would be expected when it was really for sure on. And then yeah, I would say control is like a generic term for both like negative and positive controls. They're necessary for comparison purposes when you're measuring things. And so a good experiment should have controls. It doesn't always do, but I've noticed a lot of the AP Bio questions asking about the components of like, what would make a good control or identify the control in these experiments. So this is what this question is really getting at, okay? Nice job. Okay, so let's move on and we'll take a look at our next question. So as always, I do want you to work on this on your own. So you got to be careful and read this quite carefully and go ahead and throw in the nucleotide sequence that you end up with in the chat, but don't hit enter. We'll review it together. I'll give you, we'll give you 2.5 minutes, I think, for this question. Okay, everyone, go ahead and throw in your sequence for the essentially what's going on with this question is asking for the DNA sequence of the viral RNA that is above, that was found in the host. So go ahead and tell me, what do you think? What would be your, I don't want to give it away, but yes, the DNA sequence here. Okay. Go ahead and throw in those answers. Why are they all? Oh, weird. Nick, they're all like, they're all being held for review. They're coming in, they're coming in. Oh, there we go, okay. Nice. Okay, so there's two components to this. Sometimes it's hard to remember how to do these. We're looking for the complement, but it's kind of like the opposite of how we normally do. We normally were given like a DNA sequence, and then you're asked for the transcription of it. What does it look like when it's RNA? This is because we're looking at a piece of viral RNA, and we're looking for like, what does it look like when it's been transcribed? Retroviruses do that. They reverse transcribe the RNA, make it into DNA. That's what's get inserted into the host genome. Yeah, so yeah, it's okay, Ria. Yeah, we're gonna do that together. We're gonna do this together because it is actually really easy to mess this up. Okay, so this is what I do. When you do transcription, you read from right to left. You read from 3' to 5', but what you're creating is in the 5' to 3' direction. Okay? That's okay. That's okay. We're going to do it together. So you start from the right. You start from this guy right here. And then I'm going to switch color so that it stands out a little bit more. So the complement to U is normally A. And so this is being written in the 3' direction. We're going to do this backwards. So this is A, G, C, C. And then A normally goes to U in RNA, but it's a T because we're DNA. So this is T, T, A, C, G, G, C, A, T, right? But you cannot write it like this. The correct answer cannot be written in the 3' to 5' direction, team. You can't do this. So what you need to do is flip it and then rewrite this in the 5' to 3' direction. If you write it in the 3 to 5, it will be marked wrong. So C, C, T, T, A, C, A, C, G, G, C, A, T, 3' at the end. So this would be full marks. Okay, so you need to have the correct sequence. You need to have thymines and not uracil. And then you also have to have it in the 3' to 5' direction to get the full points for this question. If you got this wrong, I want you to sit there and assess this. Like, what part of this was difficult for you? It's always in this order, Isabella. It's always going to be written as 5' to 3'. That's kind of like, giving you a piece of text and it's written like mirror-wise and saying, oh, it's still right. It's still the right sentence. Nah, nobody reads it that way. So you have to flip it and make sure it's always going from 5' to 3' from left to right. Okay. And TH, nobody would give it to you in the 3'. Even if they did, they would write it 3' to 5'. And then you look at that and think, that's ridiculous. Why would you do this? But I've never seen any text ever give you it always has to be noted on the ends, which end is which. But by convention, it's always going to be 5' on the left and then 3' on the right. No. So all it says is, Matthew, since it didn't say explain or describe or anything like that, it just said predict, all you got to do is give the answer to get the full points for this one. So Jolly, I thought you had to pair 5 to 3 and then the second strand was 3 for 5. That is correct. So you got the 5 to 3' end on the top, and when you're doing the transcription, what you're going to do is read it from right to left, but then you're writing it from the 5' end on the right and the 3' end on the left, and then you got to flip it when you're giving your answer. So Jason, great question. Coding and template strands. I mean, kind of, right? Since if we were like, pretend this was an RNA and we're just talking about like a sequence or something like that, then the one on the top would be like your template strand, and then the one on the bottom would be like, so the template strand or the antisense strand or the non-coding strand, and the one on the bottom would be like the sense strand or the there you go. Yeah, okay. how would it be different? How would the answer be different if it was a normal RNA sequence? If the question, Ria, if the question asked you what would be the DNA that it came from, that would also be like the same answer. That would be totally fair. Always 5' to 3' for DNA and RNA, Isabella. So it's ubiquitous across like nucleic acids in general. It's always 5' to 3'. Nice. Okay. Great questions. Great questions. Both for both DNA and RNA. Correct. Okay. So moving on, let's take a look at our next question. Same prompt here. So again, let's take about 1.5 minutes. So you're familiar with what's going on here. Take about 1.5 minutes, construct your responses for this, go ahead and type it in, and then we'll press enter together and take a look at your answers. 1.5 minutes here. Okay, so we've got a cell that is suspected of being infected with HIV, and we're asked to construct an experiment that is used to identify whether it is infected. So go ahead and throw in your responses for what would be a possible experiment to determine whether or not the cell does have HIV in there, probably something to do with the fact that this is a retrovirus, and then you can analyze the host genome to see, what's going on. How do you determine whether or not you got HIV or viral DNA that is in the host genome of the cell? So go ahead and throw in your responses. So I did construct this one thinking, I know there was a question about biotech, and, I did look up and see which parts of biotech are you expected to know for the AP Bio exam. So electrophoresis, PCR, things like transformation, sequencing components are all testable things that you should be aware of. You're not asked to know exactly how to do those experiments, but more about the application, like the usage of that tool. So let's see. Elise says we can use gel electrophoresis to compare the DNA of the suspected to the DNA of the normal cell with the DNA of the virus to see was successfully reverse trans. I know it's really hard to type some of these things because you can't really see all your response. I totally done that. Yeah, gel would work. Now, I like the fact that you've got controls there as well. Like you've got the non-infected cell, you got a cell that is definitely infected, and then you can compare, the kind of bands that show up. Components, it's possible to detect whether or not match with healthy. Nice, well done. Well done. Electrophoresis, yeah. Dye both the sample and the HIV, place on adjacent wells, isolating the band. So if you're looking for specific band types, yeah, I like that. So here's my possible response to this question. And it does look very, very much like you. Like a lot of you guys pulling up the electrophoresis component. I was trying to think, like, would the College Board require you to know that you should, like, you can't just run the genome by itself. It's just going to give you one giant blob. You kind of want to see like what aspects and chop it up into finer bits. But But I think just understanding, if you had the gel electrophoresis and then you had the controls, I think that would work. I think that would work for this answer. And you don't really need to be specific. It's more about like, do you understand what the tech is used for, I guess. Yeah, so Nicole, that's the other one I was thinking of. If you knew the sequence of the cell and then you're looking for specific things that are not part of the cell's normal genome, then that would also work. So that's kind of more of an issue with like sequencing, and then considering things about, you know, possibly using PCR, like just to amplify the signal. So that's what PCR is for. PCR is polymerase chain reaction used to amplify the signal so that you're not just like looking at one strand of genetic information, you're making it much, much bigger. Therefore, you're able to like actually measure it properly. That's what PCR is. Yeah. I would say for a question like this, show your knowledge of the tech, right? Describe how exactly you can use electrophoresis. The more details you can fill in there, the more chances that you'll get all the points that they're looking for. Just saying you can use electrophoresis and not actually show exactly how that works won't really give you the answer, I think. Okay, yeah, Sanger, good old Sanger and sequencing, you got it. I did add in an answer at the bottom with PCR, more about, like, so PCR is about seeing how much is there. Like, it's really hard to measure one strand of DNA. It's really hard to do really any experiment with just one strand. And that's very possible. If you're just examining a cell, it'd be really hard to see exactly if that was a true signal or not. So what PCR is used for is just to make the signal a lot bigger. That's it. Yeah, sequencing would totally be an acceptable answer, especially if you knew a sequence that was specific to the viral genome. Oh, RFLPs, Ananya. I'm impressed. That's essentially the very first answer that I've got in the restriction fragment length polymorphisms is another way of doing that exact experiments. I just didn't think that was like within scope of AP Bio, but I'm impressed that you knew it. Yeah, it's, if you describe what you just have there as a bunch of questions, that would totally work. Well done. Nice, okay. Okay, let's move on. And we've got another set of questions to go. Okay, so here is a pretty more broad question. And again, I'm going to give you 3 minutes to work through this one on your own. And then again, we'll compare some notes. So I'll see you back in 3 minutes. Okay, team, go ahead and throw in your answers. We're looking for 3 enzymes that are necessary for transcription to occur. And in addition to that, you need to describe briefly what their roles are in transcription. Oh, I'm sorry about that, TH. Yeah, no, I'll make sure to move it next time. No problem, no problem. Okay. Well done. Yeah, we've got helicase. It separates the double strands of the DNA. That makes sense for sure. We've got polymerase, which builds the new hnRNA or like just the RNA strand based off the template. That totally works too. You need, let's see, what else? Keep on putting in your answers. I know that the limit on the on the chat makes it a little bit hard to fit it all in. Good. Another one I'm thinking of. No problem. Synthase, well done. Ooh, topoisomerase is a new one. I haven't seen that one yet. Ben, well done. It reduces the, oh, good word, torque. Yeah, it reduces like the supercoiling that happens so that the DNA doesn't just like become a giant tangled mess at the end. Yeah, nice. Okay. Okay, so this is kind of like what I was thinking with a bunch of answers. Here we go. So this would be like, I would say like out of the enzymes that you need to know for the AP Bio exam, you need to know ligase, polymerase, helicase, and topoisomerase. Beyond that, you know, some of the other like more in-depth and you know, components aren't like, you don't need to know the steps for transcription. So you just need to know roughly how it works, some of the more important enzymes. Yeah, so like single-stranded binding proteins, I don't believe that is actually required for you to know, although it is definitely a very necessary component. I can't decide whether or not they would count it though. So you know what I would do is make sure that you focus on the guidelines presented by the College Board and the AP Bio Guide, and then like take a look at some of the requirements. There was an exclusion statement about the different steps, and it did say specifically these 4 enzymes are ones that you need to know. So that's like, you want to make sure that your 3 is from at least these 4 of it. Okay. Yeah. So promoters. I mean, promoters are necessary for you to get the active transcription happening, but the actual transcription process isn't necessary. You don't have to have a promoter there in order for you to get transcription to occur. Yeah, so remember, it does say specifically for transcription. Like if I was talking about, let's see, let me think. If I was talking about, let's say, replication, then DNA polymerase would be the component, right? Like to replicate from one DNA strand to get a second one, that would be DNA polymerase. RNA polymerase is what we use for the process of transcription. Nice job, okay. Yeah, they actually, there's a lot of similarity, Nguyen, between transcription and replication. A couple of the enzymes are a little bit different. Obviously the product is a bit different, but the processes between those two are very, very similar. Well done. Yeah, they are enzymes. Yeah, they are transcription factor. No, transcription factor. Oh, wait. Jason, yeah, transcription factors are not necessarily enzymes. They are usually a protein that binds. Whether or not they've got enzymatic activity, though, depends on the specific factor. Okay. I think I've got all 4 of them. DNA ligase, RNA polymerase, helicase, topoisomerase. RNA, oh, that's a good point, Flora. So you do need primase. And it kind of works the same way as replication does. But for transcription, it's funny because they actually use DNA primers, and then that's the primer that they use to start off with to get the rest of the RNA strand. And then something has to go back and replace all the thymines with uracil, because obviously it's RNA specific and not DNA. You're doing great. Okay. So here's the thing, because transcription factors are not necessarily an enzyme, and the question does say specifically enzymes that are involved in the process, I would say that because of the specificity, you want to make sure that you are matching to what the question is asking. I wouldn't necessarily say if transcription factors would work as an answer. Yeah, so there are components of ligase even in transcription. Yeah, okay. Well done, well done. Awesome job, team. Okay, let's keep on going. Oh, we're actually at time. You know what I'm going to do is just for like 1 minute, instead of giving you time to work through this, I did want a chance to kind of quickly talk about operons here, okay? So Operon is one aspect of the Operon system. They are bacterial specific. And the cool thing about Operons is that they've got a promoter and a bunch of genes after it. And all of these genes are under the control of that one promoter. So this would be an example of an Operon system. The other one that you want to know is like the Lac Operon, okay? And this is what we will classically define as a negative repressible system. In other words, its default state is going to be on. The default state of the Operon is going to be on. And I want to hear from you guys, what is necessary for there to be lots of in order for you to turn off the transcription of the structural genetic elements of the Operon? What do you need to know a lot of in order for you to turn off the Operon? So the Operon is all here to code for the components. It's a bunch of protein product enzymes that all work to code for the enzyme like tryptophan synthetase. And so it's cool. Operon systems are really cool because they've got all their built-in controls all in one. Yeah, Flora, exactly, right? And that should totally make sense. The enzyme makes tryptophan. If I have a lot of it, I don't need to make any more of my enzyme. It'll go back, bind to its co-repressor, and then shut off. So essentially, like, this is a repressor. So this is like this big protein product called a repressor, like this. But the repressor cannot do it on its own. They can't. In order for it to bind to the operator and block my RNA polymerase, RNA pol, this needs to also have tryptophan. Okay, so the repressor with the tryptophan together makes a complex, binds to the operator, and is physically blocking the RNA polymerase from moving on down and translating the TRPE, D, C, B, A, which all make the enzyme that makes tryptophan. Yeah, you got it. Okay. Co-repressor sounds just like what it is, Grace. It is a repressor that needs two parts, or at least more than one part, okay? So repressor is coded for by the TRPR gene, but it also needs the tryptophan kind of bound together, like in one thing, in order for it to bind to the operator and block transcription. Well done. Yeah, Molly, at this point, I don't quite know if we're doing more than what we have for tomorrow, but I mean, stay tuned. I can't make any promises. We are definitely talking about doing more though. And I'm excited. I do hope we get to keep on doing them. So let your friends know. And we are done officially for today. Well done, everyone. I'm impressed. And yeah, thanks for joining us. We will see you back here tomorrow. We're going to do some inheritance. I love those kinds of problems, so I'm excited to do them. Have a great afternoon, everyone, and thanks as well to Nick. I will see you tomorrow.